Go
Yes.
ahead.
Okay. Now on the behalf of Indian Phytopathological Society,
morning,
New
madam.
Delhi, welcome to all participants. Today, 20th lecture series on the of Indian Phytopathological Society Platinum Jubilee on occasion of 75th year Azadi Ka Amrit Mahotsav, organized by our world most popular plant pathological society, that is Indian Phytopathological Society, New Delhi. First of all, I would like to thank our speaker, today's speaker, Dr. Pranab Datta, Associate Professor, Plant Pathology, School of Crop Protection, College of Postgraduate Studies in Agriculture Science, Central Agriculture University, Umian, Meghalaya, India. Thank you very much, sir, for the accepting our invitations lecture on use of green engineered zinc oxide nanoparticle for better plant health management. And now I request to all participants to like and ask question in the chat box, in comment box, and subscribe the lecture in YouTube or in ongoing series. These lectures are also available in our IPS websites. And now I invite Dr. Prashant Jambulkar, Associate Professor Plant Pathology, Rani Lakshmibai, Central Agriculture University, Jhansi, for welcome our today's speaker, Pranab Datta.
Thank you, Dr. Pramod. It's been pleasure to welcome Dr. Pranab Datta. He's a very good friend of mine. And in this IPS Platinum Jubilee lecture series, as a 22nd lecture in this series. And I must tell that Dr. Pramod Datta is a very good scientist and young, energetic person. And he's a very good teacher even. So we will be having a very a platter of scientific as well as a kind of he will let us understand the nitty-gritty of the nanotechnology for the plant health management. So I will request on behalf of IPS, and I will welcome Dr. Pranab Datta for agreeing to deliver the lecture on a very short notice. So stage is yours, Dr. Pranab Datta. Please share your screen.
Dr. Pranav is not visible on my screen, or is it like that?
Dr. Pranab, please share your video. He is visible on the screen. Just talk to him. Yeah.
My slide is visible.
Yeah, slide is visible.
Yeah,
You are not
visible.
visible, sir.
Okay.
Let us.
Okay.
Please put your video on also. Yeah, slide is visible.
Okay. So, good morning to one and all.
Dr.
So
Pranab,
respected
sir, first I
resident.
introduce to yourself, sir. So just 1 minute, sir.
Okay.
Dr. Pranab Datta.
Okay.
Dr. Pranab Datta.
Yeah, sorry. We have to
Associate
introduce.
Professor, Plant Pathology, School of Crop Protection, College of Postgraduate Studies in Agriculture Science, Central Agriculture University, Umian, Meghalaya. Dr. Datta obtained MSc and PhD from Assam Agriculture University, Jorhat, Assam, India, and postdoctoral training in nanotechnology for FERA UK, served for AU, KVK West Tripuka, Krishi Vigyan Kendra West Tripura, and Central Silk Board. His research area includes biological control, nanotechnology, ecotoxicity, ecology, and plant defense. Actively involved in teaching, research, and extension activities for the last 2 decades, and published more than 100 research papers, 45 book chapters, 9 practical manuals, and he is the recipient of the DBT Overseas Associate Memorious Contribution Award in Sericulture 2007 by Central Silk Board, ICPP 2018 Bursary Award by the American Phytopathological Society, USA, and the Fellow of the Biocontrol Advance and Fellow of the Linnean Society of London's, etc., and established 2 labs on one on biopesticides production at Meghalaya and Nano Lab at Central Agricultural University, and Anand Agriculture University, respective. Dr. Datta developed 7 bioformulations, filed 5 patents, and commercialized 4 bioformulations, and pursued 16 externally funded projects of DBT, DSC, Osceola, Country, Florida, PCIL, IUS, RKVY, and BRNS, ICRNB, AIR, ICR, NCIPM, etc. And the life member of 9 professional societies, served as an editor of Indian Phytopathology, organized 3 ICR-sponsored and NAHEP-sponsored courses on biological control and biofertilizers, and a national workshop on biocontrol as its directors, and supervised 3 PhD and 11 MSc students as a major advisor. Dr. Datta, sir, very good, sir, for your this lecture. And now, sir, start the lecture, today's lecture, sir.
So warm good morning to one and all. First of all, the respected President of Indian Phytopathological Society, Dr. Pratibha Sharma Madam, then President-elect Dr. Rakesh Pandey sir, then officials of Indian Phytopathological Society, and respected senior plant pathologist of the country, and that means my dear student friends. So first of all, I'd like to thank the IPS members for selecting me to share my experiences, okay, on this zinc oxide nanoparticle. And I have chosen this topic, that is use of green engineered zinc oxide nanoparticle for better plant health management. So before going into the depth of this my presentation, first of all, let us know that what do we mean by nanotechnology. There are various agencies, they have given some very specific definition of this science. That is, they mention that it is the design, characterization, then production and application of structure, devices and system by controlling shape and size at the nano scales. So this thing, actually this nanotechnology is a combination of art and multidisciplinary science, where the scientists, they used to manipulate the matter at nano scale. That is a 1 to 100 nanometer. Well, all the properties, that is physical, chemical and biological properties, that means that change drastically. So we can take the example of, say, gold. Gold in bark material, it is actually look like, say, bright yellow color. But if we convert it to the nano scale, then the color may changes to your, say, transparent. That means color may goes to the, it become transparent one. But this science is not new. If you see the history, 2000 years ago, sulfide nanocrystals were found to be used by the Greek and Romans to dye their hairs. And if you go back to, say, 1000 years ago, if we see the fact of 1000 years ago, where gold nanoparticles of different sizes were found to be used to produce different color, that means different colors in stained glass window. But this science got momentum when actually in 1959, Richard Feynman mentioned that there is a plenty of room at the bottom. Then in 2000, in the year 2000, that means a national nanotechnology initiative has been started. And after that, the many scientists, they started working on this science and technology. And as of that, it has been found to have the wide opportunity is application, especially from medicine to healthcare, then biotechnology, material and manufacturing, energy, information technology, then environment, then cosmetics, textiles, agriculture, and the food industry, and many more. So as we are agriculturists, so we are definitely interested that means in our field where this science can be best used. So till to date, many nano product has been that means available, had been found available in the market. These are nano fertilizer, then herbicide, pesticide, then your nano scale carrier, nano sensor, veterinary care, fisheries, aquaculture, and many more. Many a times this type of nanoparticle has been found to be used in, say, your for preserving that ready to eat food or ready to make food. So now that means as we are plant pathologists, we are definitely interested how this that means we the pathologist can take the advantage of this science. So in our subject in plant pathology, this can be utilized to detect the plant pathogen, then plant disease management, and to enhance the plant defense metabolite. And my group, since last 2014, we are working on 4 different type of nanoparticle: zinc, gold, silver, and copper. And as we know, in case of nanoscience and technology, nanoparticles are considered the most important, or say basic building block for this science. And there has been while searching through this patent that means search, I have seen huge numbers of patent has already been filed. That is, this patent are related to plant health management, that is plant health material. So in today's lecture, I'm going to share some of my experiences, especially on zinc, that is zinc oxide nanoparticle. Now, question may arise is why I have chosen this zinc. As we know that US based, that is Food and Drug Administration, they have actually reported that zinc oxide compound that is as a grass material, that is called generally recognized as a safe material. Moreover, zinc oxide nanoparticle in particular, that is in particles, they are environment friendly and they offer easy fabrication and are non-toxic, biosafe, and biocompatible, making them an ideal candidate for a biological application. Moreover, these type of nanoparticle are considered as one of the most important vital nanoparticle for our say normal biological system. And beside that, these type of nanoparticles are used extensively, especially in biomedical applications such as biomedical imaging, then drug delivery, gene delivery, and biosensing of the molecule of interest. And moreover, these nanoparticle, beside having other good property, they also possess antibacterial, antiviral, and deodorizing property. So for synthesizing this type of nanoparticle, various chemical methods have been proposed by different scientists. Okay, but this suffers various that means disadvantages due to the involvement of high temperature and pressure, and the use of different type of toxic chemical. But on the other hand, this green approach are gaining interest, circumventing the high cost and use of toxic chemical and for reduction. So my team, we have actually standardized that means synthesis protocol, that means green synthesis protocol of different nanoparticles, is starting from silver, copper, gold, and titanium dioxide. And especially zinc oxide nanoparticle, we are actually for synthesis of zinc particle, we are utilizing common botanicals. Okay, especially I can take the example of classic that means work done in case of say this periwinkle. By taking the advantage of periwinkle flower, we have synthesized few zinc oxide nanoparticle, and we did some work for as a plant head material. After synthesizing this nanoparticle, we have to characterize to determine the size, shape, crystallinity, then charge, concentration, functional group, etc., which are actually done by following the standard protocol and with some sophisticated equipment like Dick centrifuge, then electron micrography like TAM, SAM, FM, then nanotrack analyzer, that means then FTIR to determine the functional group, DLS, and XRD. So during our work, we have seen that means the Zeta potential of synthesized nanoparticle, zinc oxide nanoparticle was that means satisfactory. It is called your minus within minus 20 to 25. From here, we can see our synthesized nanoparticle were a little bit stable. While doing the UV study, and where actually we tried the absorption peak up to in the range of 355 to 380 nanometer. But for our study, we have seen that means our nanoparticle, it showed the sharp peak at 376 nanometer. Then XRD performed at 2 theta range of say 20 to 80 degree at 40 kilovolt with say divergence of 10 minutes in 2 theta by theta. it actually it continuous in continuous scan mode. We performed that EDX spectrum, and that is that in the EDX spectrum depicted a strong signal of zinc and oxygen molecule. Then followed by your that is electron microscopy study. The same micrograph showed that cylindrical and hexagonal shape of zinc oxide nanoparticle with a size of 50 to 65 nanometer on an average. On the other hand, TEM micrograph showed well dispersed minute particle of agglomerated zinc oxide nanoparticle which were hexagonal with average size of 27 to 50 nanometer. And this TEM image actually shows a less intense layering at periphery of nanoparticle which could relate to the capping of extract compound on the resultant nanoparticle, that stating that means this nanoparticle of these are actually binded with some biomolecule as a your component of reduction as well as capping agent. Then we followed for we proceeded for your to determine the functional group, to study the functional group through FTIR, where we have seen that means primary and secondary alcohol bond, then carbon nitrogen bond, zinc oxide, then aromatic nitro compound and alkyl bond, and oxygen and hydrogen bond. And we that means through AFM image we could able to see that means this nanoparticle were with a pointed shape with uniform distribution on a particular surface. So as I said, we have standardized the protocol, we have determined the concentration, and this is the generalized flowchart for synthesis of nanoparticle. But at the time of synthesizing nanoparticle, we have to give more and more concentration, especially on 5-factor. Number one is pH, temperature, metal type, either fungal biomass or this plant extract, as well as incubation time. Okay, there are numbers of protocol, but we have to standardize these things based on our need. That means if our need is say to get a nanoparticle of 25 to say 50 nanometer, we have to standardize it through different trial and error method. So after synthesizing, after getting that means satisfactory result on characterization, okay, we proceeded to study their efficacy against some of the soil-borne plant pathogen. Here I have mentioned especially Rhizoctonia solani, Sclerotinia sclerotiarum, Sclerotium rhodesi, and Fusarium oxysporum, as because these organisms in our region is creating problem cultivating the crop, especially in the farmers' field. So, and moreover, say the soil-borne plant pathogen, especially Rhizoctonia, Sclerotinia, Sclerotium, rhodesi, they produce a huge that is resting structure like sclerosia. And sclerotia are very difficult to manage by application of different type of chemicals. And they can survive up to say many years. And as and when they used to get the proper environmental condition, they used to start their attack. So there is till to date, probably there is no, that means resistant variety to curb this, to face the problem of this type of that means pathogen. So we started working to standardize on a specific dose of zinc oxide nanoparticle. We tested 1 ppm concentration to 200 ppm concentration, and this result showed that from 50 ppm onwards we got some satisfactory result, but at 100 and 200 ppm concentration, the mycelial inhibition of this zinc oxide nanoparticle against this Sclerotinia sclerotium was found higher. Same result we also got in case of Rhizoctonia solani, that is from 50 ppm onwards, the result was quite satisfactory. And this result was compared with the common, that is called your recommended chemical, that is carbendazim at 2000 ppm. Then we proceeded to see their efficacy against the sclerotia, that is resting structure. We treated all this sclerotia for 5 minutes at different concentration. This is for Sclerotinia sclerotiarum. This is followed by Rhizoctonia solani. And then these treated sclerotia were placed to media to see their germinability. And we have seen that from 50 ppm onwards, the sclerotia failed to germinate in case of Sclerotinia sclerotiarum. But in case of Rhizoctonia solani, from 100 ppm, at 100 ppm, we got initially some growth, but after that, the growth was suppressed. Then at 225 ppm, there was no growth of this from this sclerotia. And up till that, we didn't know what is the actually mechanism involved. That means how actually this type of organism, this type of nanoparticle is suppressing the growth of this this type of pathogen. So we did some study, basic lab study, where we have found that zinc oxide nanoparticle, when come in contact with outer surface of plasma membrane, they disrupt the structure of the plasma membrane and changes its permeability. Then this type of disruption of the membrane structure and subsequent build up of zinc oxide nanoparticle into the cytoplasm, and they interfere with the your fundamental process of cell growth. And we also seen that zinc oxide nanoparticle inhibit the growth and interfering the cell function and causing deformation in the fungal hyphae. And as we know, this type of nanoparticle can generate your zinc ion, okay, which can that can fix to a fungal surface, and they can form ROS, resulting in an undesirable condition surrounding the fungi. Okay, and this particle also, as I said, that generate ROS, especially your hydroxyl radical, and sometimes singlet oxygen species, and in turn they stimulate the cell death. And during electron microscopy study, we have seen it can enter and it cause deformation of the whole mycelia when we expose it to, say, only for 5 minutes at 100 and 225 ppm concentration. Then we did some work on that means method of application. We tried 5 different method of application: seed treatment, soil application, injection, seedling root dip treatment. Okay, and I actually applied this nanoparticle at different doses, where we have seen that means out of all this method, foliar method was the best to that means to establish or to make entry of this type of nanoparticle within the plant cell. This may be due to presence of stomata, or say hydathoth, or the lenticels, or say owned lenticels in the fruits, or say stems, than the owns that are present in, say, your plant body. And through this, probably they could able to move or say translocate in different plant parts. Now again, question arises, when they will enter in the host cell, whether there will be any effect on the cell component. So we did some confocal microscopy study where we have seen they can enter, but there was effect on the cell component, especially protoplasm, protoplast, and other component like your mitochondria. Then we also seen that this type of nanoparticle at certain concentration, they could able to enhance your major secondary metabolite like phenol, flavonoid, and terpenoid at certain concentration. But when we cross this around 225 ppm concentration, when actually we treated our plant material with 225 ppm concentration, this that means it has some bad effect. It also have the ability to enhance the nutrient uptake ability, plant growth, and yield attributing character, which we established through a series of in vitro as well as your glasshouse test. Then we proceeded to study on karyomorphology, especially as well as biochemical changes if we treat a particular plant material with this type of nanoparticle. So here in this study, we actually took observations on total chromosome length, then volume of chromosome, arm ratio, relative length of chromosome, centromere index, total form percentage. These were observed for cytological and karyomorphological analysis. And in case of biochemical analysis, as I said already, that we studied on the flavonoid, alkaloid, phenol, TSS, total chlorophyll, and total soluble. From this experiment, actually, we can say this nanoparticle, as they do not impose any negative impact on the plant at certain concentration, and also have the potential to increase the higher production of few secondary major metabolite in the plant, which will be beneficial to health and also beneficial to the plant growth. These are some of the photograph of microphotomosome at the metaphysics stage of say of the sample which were treated with your zinc oxide nanoparticle by different process. These are some of the camera lucida diagram of metaphysics stage of different treatments. Then these are some karyotypic and ideodiagram photograph of soil treatment, then foliar spray, root dip treatment, injection, as well as control, where we have not seen any drastic changes in comparison to the control. So We also studied their efficacy, as I said, at different concentration, especially 50, 100, and 225 concentration as a seed treating agent. Here we have seen up to 100 ppm concentration growth was good, but at 225 ppm concentration, sometimes we used to see curling or say epinasty or hyponasty type of symptom. Even at some concentration that is up to 100 ppm, root biomass were good, but beyond that, some inhibitory effects we have observed. Interestingly, we have observed around 22.76% of increase in your nodulation in say leguminous crop like your French bean. But what is the science behind it? What is the mechanism behind it? We don't know. So the works on the gene that is regulating this nodulation in leguminous crop that is going on. So in the coming days, probably we say many things that means what is the mechanism behind this increase of nodulation in say leguminous crop, especially green gram as well as French bean. So these materials were taken by some scientists of horticulture department of Assam Agriculture University. They tested to see their effect on the best life of your cut flower, and they reported that they could able to get higher base life, especially in case of rose, lilium, and gerbera, up to say 7 days. So then we did some work where we have seen that increased base life, that means that is coincide with the higher level of your endogenous proline and lower level of superoxide radicle and a higher activity of PDAs in the treated pot, and that's why probably they got higher base life. Then we did another work on that means shelf life of banana. That is a big problem because after harvesting of this type of banana, and when that means the disease like crown rot and anthracnose is big problem, especially jahazi, that is Grand 9 variety. So through a student research work, we have applied zinc oxide nanoparticle 7 days ahead of harvesting. Then after harvesting, these bunches were that means dip in the zinc oxide nanoparticle at 100 ppm concentration. Then by doing that, we could able to reduce the incidence of crown rot and anthracnose up to 38.6%, and the shelf life we got a little bit 3 to 4 days as compared to the control. Then beside that, in collaboration with the scientists of National Seed Project, that means another work was done on that means the use of this type of nanoparticle as a nanopriming agent, especially for the chickpea. As we know that for chickpea, the storage is a big problem. After 3 to 4 months, that means viability they used to that means it goes down. So we used zinc oxide nanoparticle as a priming agent for 6 hours, followed by that means drying for say drying so that moisture percentage comes down to 9 to 10%. Then we analyze through that means different biochemical parameters and some physiological parameter, because these seeds after that at an interval of 2 months we used to grow in the pots. So here we have seen especially for the biochemical parameters, the peroxidase activity was found increase with the increase of storage time, but the rate of increase was low in say primed seed, which indicate that probably due to the priming, the firmness of the seed increases, resulting more storage life. Interestingly, we got around 8 to 9 months storage life. And this seed, when we tested against Fusarium, Oxysporum, it showed very good result. Good plant is done in say pot conditions. Then while doing work on alpha amylase activity, it was found the highest in say prime seed, but decreased with the increase of the storage, but the rate of decrease was lowest in the prime seed. We also got highest protein and the lowest TSS in the prime seed than the control. And beside that, as I said, the physiological parameter from seedling germination to seedling vigor index, we got a satisfactory result as compared to the control. So as we know, this zinc oxide nanoparticle have UV trapping ability. And at the time of application of entomopathogenic fungus and many a time biocontrol agent like Trichoderma hergenium in case of field, okay, due to this UV rays of the sunlight, we many a times we used to get the erratic result. So we tried to see that compatibility of this type of biocontrol agent, especially Metarhizium encephalii, Beauveria bassiana, Verticillium leekani, and Paecilomyces liliaceanus, and some of the organisms that is antagonists like Trichoderma, species like Trichoderma hergenium. Here we tried to see their compatibility at different concentration, and we observed the mycelial growth, biomass, and biochemical changes on the TSS and protein. Here we have seen, beside that, we also tested their compatibility with Bacillus thuringiensis at different concentration. Here we observed that, especially on that means on biochemical changes, in case of say fungal mycelia, mostly your antagonist and your entomopathogenic fungus, we have seen upon exposure to different concentration of the zinc oxide nanoparticle, it showed decline in the content of both total soluble sugar and total soluble protein. And when we tested the concentration of beyond say 150 concentration, but up to 100 it was good. Then this TSS was also found to be declined, but BT was found to resistant to all the tested doses. And some nematologists of our nematology department of and Assam Agriculture University, they tested at this concentration and they reported that after 72 hours of its exposure, the J2 stage of Melodogyne incognita, that means they die up to say that means percent mortality they have recorded as 100%. And at 100 ppm they recorded around more than 93%. But what is the mechanism behind that thing we have to study in the coming days? Now, always we used to face one question, whether this type of nanoparticle have any toxic effect to these human cells. So we started working on the mammalian toxicity by taking the cell of your African green monkey, where we concentrated on especially that means this type of hero cells were exposed to different concentration of nanoparticle, especially 50, 100, and 200. And we exposed this type of nanoparticle for different hour, 1 hour, 4 hour, and 8 hours. And this result exhibited that higher cell internalization of zinc oxide nanoparticle by this hero cell was observed even at say 1 hour of its exposure, but it is dose dependent manner. I mean to say, with the increase of dose, was more. And at 220 200 ppm concentration, we got around 89% cell internalization. Again, with the increase of time of exposure, that is from say 1 hour to 4 hour and 8 hours. So with the increase of time of exposure, the uptake was found to be increased with the this increase of concentration. Beside that, we did this MTT test, MTT assay, which is a colorimetric assay for a soil metabolic activity. Here, the results showed that the viability of cells at 50 ppm is quite satisfactory. That is 77.01%. But it was found to decline to 69% at 200 ppm. So this type of result indicated that there is a mild effect on mitochondrial metabolism of hero cell, even at the highest concentration, that is 200 ppm. This was followed by your lactate dehydrogenase assay. Actually, this is considered as one of the classic cytotoxic assay, which is used to assess the level of plasma membrane damage in a cell populations. So here, again, we have observed that means the cytotoxicity of zinc oxide nanoparticle increased in a concentration dependent manner. But the percent cytotoxicity was recorded very less, that is 12.31% at 50 ppm, but it and highest at say 200 ppm concentration, that is 23.28%. So thus we can say there is a low toxicity effect of biosynthesized zinc oxide nanoparticle in hero cells. So overall, I want to say that zinc oxide nanoparticle, through our study, we can say that the single molecule, single particle which have the ability to suppress the growth of soil-borne plant pathogen, increase the secondary metabolite. They can have the ability to cause mortality of the root knot nematode, increase the nodulation, enhance plant growth parameter. And our studies showed that there is no adverse effect on the host up to 100 ppm concentration. So this type of work we have already published in few journals like Applied Polymer, Journal of Photochemistry and Biology, Journal of Nanoscience and Technology. Then one work especially on that is priming agent of zinc oxide nanoparticle and silver nanoparticle, recently published in Journal of Agricultural Science and Technology from Turkey. And also in our prestigious journal, that is Indian Phytopathology, very recently we published 2 work related to your silver nanoparticle as well as chitin nanoparticle. So these are few of the product which we have prepared through that means by mediating microbes, then by mediating some botanicals, and by adopting some chemical approaches. So beside that, respected participant, so in the future to do more and more work on this the UV protectability of this type of nanoparticles, so that we can use it as a component of a bioformulation, so that in the field level we can get very good result of, especially for this entomopathogenic fungus. Then there is a very less literature on that means on how this type of nanoparticle interact with the environmental matrix. Then this is not understood well. That means how these nanoparticles they distribute through the plant vascular system. So many more such work we have planned in the coming days. Probably we can give more and more information to research. So let us start from the very small, and I would like to take the opportunity to acknowledge the contribution made by my student. So first row, these are the PhD students who also helped me to get a lot of result in not only in nanoscience, especially more particularly in say biological control. So and a few students, they came from different institutes, say NIT, then different say national as well as regional college, even from USA. They came and joined to my lab to learn the technique of especially nanotechnology as well as bioformulations. And now they are actually acting as a good resource person for transferring the knowledge, okay, of nanotechnology and biological control in other part of the country or say foreign countries. So I would like to thank the organizers, especially the this fraternity of Indian Phytopathological Society, and I would like to sincerely thank all the funding agencies, starting from DST, my present authority, then DBT, ICR, Central University, and more particularly my my dear student friends. So thank you all. With this, I would like to conclude here.
Thank you very much, Dr. Pranav Datta, for vibrant lectures. It will serve to our participants working in the field of managing plant diseases and nanotechnology, and extension workers, research, and policy makers who will directly involve in nanotechnology in our country. Vibrant lectures are on the use of green engineered zinc oxide nanoparticles for better plant health management. And then now I invite to our participants for the open discussions or questions to the Dr. Pranav Datta. Participants.
Dr. Pranav, please put on your video. Let us see. Thank you.
Okay. Thank you once again to Pratibha Madam, then Rakesh Pandey sir.
Yeah.
And I have
Thank
seen
you.
a lot of senior people here.
Let me have one thing.
Lot of
Because
senior
Pranav is doing very good work. Not from today. I have heard his lecture, and he, first of all, I congratulate him for doing such a wonderful and outstanding work in the nanotechnology. So I have one clarification, Pranav, because I work also on the nanoparticle. Yes, the size of nanoparticle become very important for managing disease, because when we are having a smaller size, they are having very good anti-aging and health-related, human health-related activity. And when we are having the size of the nanoparticle longer, then it is antibacterial, antifungal, and nematicidal. Because with the nematodes I have worked, and I have seen the size of the nanoparticle play a very important role. So I think you include this thing also. This is my just a suggestion. You include the size of
Yes.
nanoparticle in your future study. It will give some more output and some more technology in your science. As I worked on the nanoparticle with the Sinorhabditis elegans.
Okay, okay. Thank you, sir.
So,
Actually,
thank you.
for,
Great lecture. I enjoyed.
for our cases, thank you, sir. So for our case, what happened? Most of the cases we try to synthesize nanoparticle from say 30 to 55 nanometer. But for say, sir, for chitosan nanoparticle, we could not able to do that one. We tried a lot. We did lot of that means tried. That means a lot of the experiment, but we failed to synthesize chitosan nanoparticle below 100 ppm concentration.
Yeah,
So it's okay, below 100 nanometer size. Yeah, but for silver, yours are this zinc oxide we could able to synthesize from 30 to 67 nanometer. So, but sir, more work we have to do. We have to devote more
no, that's
time.
congratulations.
Then
You
and
have worked a
then
lot.
only probably there will be some. Thank you, sir.
Yes, congratulations. You are working very well. Outstanding.
Thank you. Thank you. हाँ sir, Vishwas sir.
Dr. Pranab, I am very much pleased and for your excellent lecture and your excellent contribution in nano technology, particularly not in Northeast, also for India. You are known that Pranab Dutt is working on nanotechnology and management of plant diseases. But now I have one question to you. How we can utilize this nanoparticle for management of plant virus diseases? Is it possible to manage virus diseases, because virus is nanoparticle and your items also is nano? So how we can utilize this nanoparticle for management of plant virus diseases?
So that is a very critical because
प्रणव आप वीडियो आप बंद करो वीडियो, क्योंकि वीडियो के लिए आपका प्रॉब्लम हो रहा है. हाँ हाँ अभी बताओ.
So
ना वो भी नहीं है, वो भी नहीं है.
okay.
हाँ बताओ.
Here what my experience is if we
Hello, Pranab.
I think problem in network.
Yes. Dr. Pranav?
can utilize this type of nanoparticle to activate the defense metabolite of
Any
Pranav, it's not audible.
Prashant?
Yes, sir.
I am audible to you?
Yeah, you are audible to
but
sir, but
we have some problem, network problem.
Yeah.
Anyhow, he has finished his
I think
lecture.
there is a problem with
That is
Dr.
lucky.
Pranav.
He has
Dr.
finished
Pranav.
at this lecture.
Dr. Prashant, we can wind up then if it is like this.
Yeah.
Okay, ma'am. I think
It's not audible.
Pranav also could have start mobile also side by side. I am doing like that. Mobile and computer both. So
You are very expert, Kajal. I think we should take question, no problem. And then we can pass the Pranav if there is network problem.
Yeah, he
So-
just, I just called to him. He told that there is some network issue in that area. He just
Yeah.
got message, some WhatsApp message, or so he may not take
We can take
He's trying
question.
to
If
connect.
somebody has some question, we can hear their question and if possible, we can answer. Otherwise, we can pass to Pranav.
Yes.
You can ask Prashant.
From chat box, Prashant, can you collect the questions from chat box?
Yeah. I have taken the screenshot of it.
Yeah. That can be sent to him.
Gogoja, namaste.
Namaste. Namaste.
Sir, we will convey the questions to him, and
Now I have one question to Pandey sir. Pandey
Yeah,
sir,
please.
have you any idea the management of virus disease through nanoparticle?
I think this size, as I told, we can take nanoparticle because we have not got any result till date. I don't have any experiment to see the nanoparticle impact on the viral diseases. But why not you should try at your place?
Yes.
The different
because,
size of the nanoparticle, even larger size, they will help to manage the viral diseases.
but larger size.
Larger size because smaller size, they are good for health. For example, they are anti-aging, anti-Alzheimer, anti-Parkinson. When size is less, they are having very good result. But when size is larger, they have toxicity effect. So that toxicity we can have against the pathogen. So you have to take from Sigma and other agencies of different size of nanoparticle and you can test it.
But
It will help you.
toxicity, sir, toxicity related to bacteria and fungus, but how it will work against virus?
You are the man of virology, you know better. You can
No.
see it. Because
Because
I
some
work with the
result-
fungi,
some
bacteria, and nematode.
Some report we have got in the human pathology.
Yeah, he's trying to
Pathology,
connect from
the
mobile.
result is sporadic, not clear.
Yes.
So we'll
But
wait
we
for
can
1 or
try.
2 minutes. He's trying to connect through mobile cam.
Okay, good.
Yeah, this
Just
is
connected.
Sridhar from Chennai. Can I make
Yes, sir,
a
yes,
small
please.
comment in relation to the discussion that is going on? I think we are trying to imagine the management of viruses directly by nanoparticles, as we have experienced with the other pathogens, macropathogens. But when we talk about the management of virus diseases, management of virus diseases is possible by the molecular and genetic engineering also, where the nanoparticles have got a greater potentiality. For example, in gene delivery, or if we know the virus disease syndrome very well, probably we may be able to knock out and knock in the genes using the nanoparticles, where their exceptional benefit and effectiveness have been proved in several things. So the virus disease management has to be approached in that direction, rather than expecting us to interaction of nanoparticles directly with the virus for defeating the virus.
Yes, indirect manner.
Yeah, you are very right. You are very right, sir.
Yeah, I have one question, means query to the August audience also.
Audience
Sir,
or Pranav?
as Pranav has
Audience or
said
Pranav?
that- Yes, sir.
Question to Pranav, now audience.
As an audience, we can discuss. You are also learned audience. He told that zinc oxide is compatible with the trichoderma. So can we coat zinc with the trichoderma spores using some kind of biodegradable polymers? so that we can get the benefit of both zinc as well as the trichoderma to manage the disease and all.
Prashant,
Yes, sir.
Prashant, you have to take care about the size. As I
Yeah.
told you, when
Uh-huh.
we are having larger size, it is
Mhm.
toxic. But when we are having smaller size, they are non-toxic.
They are not toxic.
So that you have to take care when you are working with trichoderma. Like Pranav has told that particular size of the nanoparticle, ZNO nanoparticle is working very good for the growth of the trichoderma.
Yes.
So that nanoparticle you can take, but you have to take care of the size.
Size.
Because when size is smaller, it is non-toxic.
Okay, okay. So we can make the reduce the concentration of zinc oxide as well as we will be having the combo of trichoderma effect as well as zinc. So to reduce the toxicity, we can reduce the concentration of zinc so that it should not be toxic.
Maybe.
Hello, Dr. Jambalkar.
Yes.
Yes.
Yeah, you can hear. Oh, Pranav. So we are glad to that you are back. We can have questions.
Yeah, number one, that is you asked one thing now that
Yes.
whether can you combine zinc oxide nanoparticle with trichoderma spores? Okay,
Mhm.
yes, because as already Pandey sir told that when the size will go beyond say it is more than say 200 or 250 nanometer, this creates problem to this that means even to the human mammalian cell.
Okay. Is it? Your voice was gone. You're not audible, Pranav. As he said that it is toxic if we reach to the size more than 200 or 250 nano. Listen.
Prashant, you can ask with the phone that there's a lot of question about the nanoparticle on viral diseases.
Yes.
Okay. Nayan Adhikari also has asked, and our great Kajal has asked. So you
Yes,
can ask this question from the Pranav. I think he will answer properly.
Network is also problem. That's why similar na network.
Okay, to activate your there is called defense metabolite of the plant. Okay, and the plant may have the ability to resist the attack of virus. So this type of literature are available.
Is it audible, Kajal sir?
Yeah, it's audible. It's audible.
Yeah, we can have a question and answer here.
Yes.
So I want to say something about with Kajal sir's question. Actually, me and Kajal sir, we are discussing on this topic since long, last 1 or 2 years. So while searching some literature, I have seen many people are trying to utilize this type of nanoparticle against your viral diseases. So from that literature, I could able to understand that this nanoparticle at say small size, if we can use it to activate the defense metabolite of the plant, which has the ability to fight against the virus, then and then only the plant will, then and then only this viral disease can be managed. But still, that work is in the nascent stage. We have to do more and more work for utilization of nanoparticle for viral disease management.
Thank you. Can we have another question? Sonali Pardia has some question.
Okay.
Good morning, sir. Sir, my question is that what Dr. Datta said that he synthesized the nanoparticle with zeta potential, negative zeta potential. But can we use the nanoparticle with positive zeta potential for the same use?
Oh, yes, that is a good question. Actually, you see, a particular nanoparticle, especially think of the nanoparticle, they should have the data potential within plus and minus 20. Okay, so within that range, if you get the data potential value, then these are said to be very stable. Okay, it may be plus 20, it may be minus 20. So that's why probably I have mentioned. Sorry, that's why I have mentioned the data potential should be within the range of this plus minus this.
Okay. Sir, I have one more question. Sir, do you think that nanoparticle treated plants have any threat on the food consumers? The health of food
I did not get your
consumers?
question. Please.
Any harm to food consumers, nanoparticle?
Oh, yes, but initially I said that already it has been declared as a biocompatible molecule. Okay, and this zinc is an important component for our cell growth of the human being. So here what happened, the research group said that it may go to the end product, which is called your say foods or say any other eatable part, but up to certain concentration do not have any harmful effect. And already I see these are actually biocompatible with the human cell. And that study already we did that up to some concentration, these do not have any toxic effect that means on the mammalian cell.
Sir, but with time, the nanoparticle may get bioaccumulated in the organism, in the food consumers. So that may have a threat on their health. Sir, what do you think on that?
So at a higher concentration, there may be problem. Already repeatedly I am telling you, but at certain concentration, because we can utilize that. Body can also detoxify the toxic effect of many materials. Okay.
Okay. Thank you, sir.
But Sonali, when we are having the smaller side, it is good for the health. It have anti-aging activities, and it has been proved in so many animal models.
Yes, yes.
Okay.
Dr. Ravindra Kumar, please. So unmute, please.
Okay, sir. Okay, sir. Greeting from IIWBR, Karnal. I have two queries, sir. Since I am new to nanotechnology field, I wish to know about the effect of nanoparticles, different nanoparticles on seed-borne fungal pathogens. My first question is this. And second one, can we use these nanoparticles for enhanced storability in case of wheat and barley crops? These are two queries, sir.
Dr. Pranav, did you get the question?
No. It is actually
First question is effect of nanoparticles on fungal
seed-borne pathogens.
Seed-borne pathogens. And another is on fungal seed-borne pathogens. And another is use of nanoparticle on use of storability of wheat and barley. Effect.
What was the question?
Question is can we use nanoparticles to increase the storability of wheat and barley?
So that experience I don't have, but I did work on chickpea. Okay, as in the chickpea we got the good result, especially on your physicochemical parameter, physiological parameter and biochemical parameter. By taking the advantage of those information, we can try. Okay, there is a possibility. Probably there is a possibility. But as I did the work in that wheat and barley, so I will not answer this question because. Whatever the experience I do have on chickpea, that information our scientists can utilize. Okay. And another what was the another question?
The effect of nanoparticle on seed-borne pathogens.
Seed-borne pathogens.
Yes. Seed-borne.
Yeah, this is already many work has been done on different type of fungus and bacteria where not only zinc oxide, even gold and silver nanoparticle were also found to be very effective. Okay. Okay.
Thank you.
When nanoparticles are found to be effective against many bacterial pathogens.
Hello, this is Gopi from Kannur.
Yes.
I'm
Gopi.
audible? Yeah.
Yeah, you are audible.
Especially related to earlier question, I can say, so if the peroxidase activity, then alpha amylase activity, that means we get some satisfactory result. Then, and especially on the protein and the TSS of that seed, okay, that study if we do, especially on the wheat and barley, and we get some satisfactory result, especially as I say, in our case, we got this peroxidase activity. That means increase with the increase of storage time. What happened here? With the increase of time of storage, the rate of increase of peroxidase activity was found to less. That means from here we came to a conclusion that probably because of priming, okay, the seed firmness, the firmness of the seed it increases. That's why probably it results in increasing the storage time. Besides that, I already mentioned that alpha amylase activity, which was found in chickpea, actually, which were primed with your nanoparticles. Okay, but this alpha amylase activity was found to decrease with the increase in the storage life. Here also one interesting point we have observed that the rate of decrease of alpha amylase activity was the lowest in the prime seed. Okay, similarly, we got very good result in case of highest protein content in the prime seed with the lowest total soluble sugar. Okay, so if that means total soluble sugar concentration increases, then the literature seed they actually lose their viability. So from this thing, we can come to a conclusion that this type of actually nanoparticle can be utilized for other crop also. Okay, actually this was an assignment given by National Seed Project, one scientist from National Seed Project, and interestingly we got this information. Okay, now this work is going on in around 4 different centers of under the National Seed Project of our country. So in the coming days, probably we can say many things.
That's
Is
good.
it okay?
That's good. Yeah, Dr. Ravindra, you might have got the answer.
Thank you so much.
And Dr. Gopi.
Dr. Prashant, I have some answer regarding Noyan's question. Noyan.
Please, sir.
Noyan has asked what is the some examples of viral disease which is managed through nanoparticle. For him, I am telling Noyan, are you listening? Noyan, are you listening?
Yes, sir. I'm listening, sir.
Some Sanhem rosette mosaic virus, tomato mosaic virus, potato virus Y, then tomato yellow leaf curl virus, barley yellow mosaic virus, some virus experiment experiment already done many scientists, and symptom development and other things, disease incidence, etc. are reduced. But full control in field level still not available. But we can now start. You are also working in Northeast and you are virologist. Then you contact with Pranav and try to use some AG, silver nano particles are effective against this virus, particularly CMV and PVY available in your area. Now you can try for management of these viruses.
Okay, sir. So nanoparticles is effective for both RNA and DNA viruses.
Because mechanism still not clear, but what is the mechanism? But that's why the nanoparticle used in plant virus disease management not show because until unless we will not do the what is the mechanism, we cannot continue. No, that is the reason. But in fungal disease and bacterial disease, mechanism is available. How it is working and But for virus the DNA or RNA. Nah, you have to control the DNA RNA replication. So that DNA and RNA. That is the my thing. Still, mechanism, the control of virus through nanoparticle, not clearly known. That is the thing.
Okay, sir. Okay.
You're right, sir. Yes.
Dr. Gopi, you are having one question.
Yeah. So Pranav Datta, your lecture was very good. I think you are doing remarkable work. But the thing is that, like, what is the stand of government of India and nanotechnology, especially in agriculture? And other thing is that, like, what is the disposal mechanism? Because we don't know what is the behavior of the nano particles actually in the environment. So suppose if we use any nano formulation, so how we can even for research also, I think it is not allowed in the field level. So if at all you do, how will you dispose? So these are the questions I have.
Yeah, for that actually, on GBT, they have formulated some guideline. That guideline they have published in 2018. Okay, so in that guideline, everything has been mentioned very clearly. So if we go through that guideline, then we can learn many things. So I request you please go through that guideline. And regarding that, your question disposal, that is a big question. Already we know that zinc oxide nanoparticle has been used in, say, toothpaste, then paint, then your sunscreen lotion, then cream. Okay, where actually the cream with that is called having the sun protection factor, UV protection factor. So by actually from there also it is going to the environment. But you see, up to certain concentration, also if it goes beyond some permissible limit, it will create some problem in the coming days, because zinc is that is, they have a high tendency of agglomeration. When this type of nanoparticles will go to the environment, then the agglomeration will be there, the size will be increased. So as already Pandey sir said, when that means the size will increase, but it may be harmful for that is called your soil microflora, fauna, and many more. So that's why I have given one slide. We don't know how it will behave when it will come in contact with the environmental matrix. Okay, you see, already actually this science, though we are working, but still in many cases it is the science is still in the nascent stage. So in the coming days, probably we can say many things, but we have to do more and more work in this line. Okay.
Dr. Gopi?
Yeah, okay, thank you.
Dr. Pranav?
Dr. Kiran?
It is correct, nanoparticle is not allowed in field level?
No, actually what happened for few fertilizers, okay, that is released by IFCO, we are utilizing. They have they got certificate from the ICR. Even one company from West Bengal, that is Camptech, they are also utilizing this type of nanoparticle prepared from hydrogen peroxide. They also got the certificate from the ICR. So we can say this is not permissible. Okay, this is not permitted.
Okay.
Dr. Kiran.
Sir, good morning, sir. Sir, can we convert the available fungicides into nano formulations, sir, in the market?
Yeah, this is another area. This is called, one technique is there. This is called ball milling. So ball milling process, we can convert macromolecule to say nanoparticle. That can be done. Many people are working in this line. Okay, that means through that process, there is a mechanical process. That mechanical process, we can convert this type of nanoparticle to macromolecule to or say macromolecule to a nano form, and it can be utilized. In that case, probably we have to be very much careful at the time of determining size, shape, sterility, then your especially your tone. Okay, because any time in the market also the people they are selling some particular in the name of nano. but I have very bitter experience while utilizing titanium dioxide from a company. They mentioned it is at 25 nanometer, but at the time of analysis, I had seen the size is beyond 167 nanometer. So we should avoid this type of thing. So before telling any say fungicide or converting any telling any material as a nanoparticle, we have to be sure whether these are actually in the nanoform or not. Okay, and through ball milling or other mechanical processes, we can convert it.
Sir, by using the different polymers, can we increase the shelf life or efficiency of the fungicides present in the market?
I'm sorry, that thing I don't have any idea, especially on the fungicide and whether we can use such as polymer or to increase the shelf life. Because fungicide, most of the fungicide, they are having the sufficient shelf life now.
Sir, for example, we will be having, except for tebuconazole, only shelf life is for 15 days. Sir. For seed-borne pathogens, diseases are inciting beyond 25 days. Sir. So, if we increase the shelf life of the fungicide, it could help the farmers. In that way, this question was asked, sir.
It is not shelf life; it is residue layer.
Sir, yes, sir, residual effect. Sorry, sir. Sir, okay, sir. Thank you, sir.
Okay.
Thank
Now
you.
I think very good discussions during this lectures due to network issue in Union. I think this lecture also available in YouTube. If any questions, you write direct in the YouTube. And now invite to our secretary, sir, Dr. Robin Gogoi, sir, for the vote of thanks to Dr. Pranav Datta, sir. Robin, sir.
Thank you, Dr. Pramod. Actually, this was not my turn anyway, because you are the very good coordinators yourself, and Prasant coordinating very nicely this lecture. So I must appreciate both of you along with the other team members. So from the headquarters, so I must thank Dr. Pranav Datta. He has actually gathered lots of information and also knowledge related to this nanotechnology. So he has been associated with this line of work for last 8 to 9 years. So I know his this capability. So based on his experience, he has delivered very fruitful lecture today. So on behalf of our society, so I profusely thank him. And also one more this thing also, he needs praise because he is the zonal counselor of Northeastern chapter of Indian Phytopathological Society. So very much busy. So here I remind that he sincerely put effort to organize this Northeastern chapter of this zonal chapter, is a national symposium. This in physical mode during the time this 6th and 7th of January this month. But because of some of the COVID protocols and SOPs, this declared by the government of Meghalaya and also our country, so that could not be materialized. But we are hopeful that within few days the situation will be normal, and that time, so definitely we'll get opportunity to have that function in a physical mode. So, and also, and a large number of participation today I could see. So at one point of time, it reached up to 58. So that way, so by seeing the title of lecture of Dr. Pranav Datta, so everybody was fascinated to attend this lecture. And I hope from his lecture, everybody is benefited. So I thank all of you, and so hope you will be participating in the coming lecture series also. So thank you, Dr. Prasant, Dr. Pramod, and here I could see Dr. Rakesh Pandey, our president-elect, as well as our present president, Dr. Pratibha Madam. So I thank all of you once again. So happy weekend.
Thank you. Thank you, Dr. Pranav.
Thank you, sir. Thank you,
Thank you, sir.
Thank you.